So I'll be discussing about some of our recent some of our development on the design of organocatalyst, mostly on nucleophilic organocatalyst, and then on the photocatalytic reductive transformation by developing some organic photocatalyst. All right. We know the incorporation of such functional group like halogens, nitro, SCF3, SPH on alkene or arene are very important because they are commonly occurring in many drugs and also found in several natural products. If we think about in halosaxonamide reagent, how we activate? One way is we just add Lewis acid, which simply coordinate to this carbonyl, making this Ne for electrophile actually weak. E can be anything, actually one of them. And then another way, base can plug this electrophile, making it more reactive, and it is easy to deliver on the alkane. There are some problem like nitration. This is not much known, and also as like 1,2-dihalofunctionalization is also very challenging. What we wanted to do is develop organocatalyst which like nucleophilic mostly, and if we have nucleophile, it can trap the electrophile to do several such transformation. I'll just discuss elaborate in the coming few minutes. If we check the reported method, like reported catalysts, there are several catalysts. Okay, they are of course fantastic. They have different activation mode. They work mostly with ionic bonding or hydrogen bonding interactions. So what we wanted to do, we want to come up with a new catalytic mode or activation mode. If we check the reported method, let's say you are doing asymmetric halogenation, there is one problem. What is the problem? Once the halogen got transferred to alkene, these halonium ion, it can undergo exchange among them. So when they undergo exchange, you lose the enantiomer selectivity. Of course, that is our end goal, to develop asymmetric new chiral catalyst. So the weak interaction is also issue, and catalytic stability is also problem, like when you have a phosphine type and they sometimes melee. And if we can develop phase transfer catalyst also in a solvent like heptane, that will be really fantastic. That was how we started our project in maybe 5-6 years before. And then you can see there, like what we want to do, we want to develop a new activation mode using a like this alpha effect. Okay, so if we check the alpha effect containing functional group, it is peroxide, hypochlorous acid. We know these two are oxidant. This is not a good for us active site. We can have the disulfide, but they are relatively unstable. What we choose is the hydroxylamine and hydrazide, and these are very common, like very cheap. You can have them in bottles. So there are NN bond and NO bond. They are relatively stable. They are unreactive toward the electrophiles, and they can generate the neutral active species, not the ionic one, and the covalent bonding are possible. So how it is working? You know, this lone pair and this lone pair, they repel each other and making them a very good nucleophile. So if we can put on the active side, it will be really a beautiful catalyst. And such active side are not known, and of course conceptually it will be novel. So when we design, and what we did, and when we just simply took these catalysts like with NHOMe, this one, and treated with NBS, it's just a spot to spot conversion. Means you add it and it plug this bromine and you form this, and this is really highly reactive. So what happens if you here actually this is what happening, it forms this, then it take the protons and we get this neutral and covalently bonding. Now imagine if we have a chiral scaffold around, it's a covalent bonding, the delivery of bromine in a asymmetric fashion is very much possible. So we have also checked by 1H NMR analysis about the like NH signal is disappearing and this is appearing, and we can also do several recycling. I'll come back in few minutes. Then when we choose, like took this catalyst with NBS, 5 mole % catalyst we choose, and we could do nearly nice transformation with bromolactonization. So when there is a alpha effect like this one, we have 94% yield in 3 hours. But when there is a no alpha effect, no trace yield. Okay, so the alpha effect playing the role, and how it is playing the role, if we know the amide, okay, this is much more stabilized. So ground state is much stabilized compared to the alpha effect. So this ground state destabilization is making this more energetic. So, and also when it deliver one of the lone pair to the electrophile, it also make the transition state much more stabilized. So the activation energy will be much less. So this is how it is doing the catalysis. And of course we have several parameters to change, like we put the sulfone, we found is a good one. Of course this some other acid we are working on at the moment, and we can put here some groups to make it more soluble, and the reaction work in heptane only, so that is really nice, and this is very lipophilic solvent. By using this catalyst we can do several transformations like bromolactonization. You can see the background reaction is trace. Each of the cases we have also conducted the background reaction, means in the absence of any catalyst. So as shown here, like 83% when there is no alpha effect is not working. With aromatic group, with phenyl group, it all work. Bromoetherification, bromoamination, dearomatic cyclization, all are going well, and also arene ring halogenation. We have total 6 reactions spanning over 50 examples, so proof of concept is already there. With this we thought, okay, can we just generate in stoichiometric amount, okay, and then we transfer this. There will be one bromine, sorry, it is like I have to change it. There will be bromine. So bromination working, we isolated and again recycle just to check the catalyst stability, and after few cycle also this reaction work quite well. So as you see here, like in absence of catalyst there is no reactions, but when you add catalyst it slowly gradually gives us the product, and there is always some induction time. We still don't know what actually happening with the catalyst in this during this. However, it is of course not decomposing because we are isolating and doing it again and again. So we need some induction time depending on the catalyst. So now this catalyst recently used for the total synthesis also, like you can see here the yields are not good. When they use our catalyst they could do nice this transformation. I just cut because to show that they have used it, and for the total synthesis of this natural product it just came. So this catalyst is quite efficient. Now we wanted to do the not bromination, halogenation, that means particularly chlorination. We know the chlorine containing drugs, there are many, and not only that, the chlorine attached to the sp2 ring is more than the other in the marketed drugs. There are several drugs I have noted, I am not taking their names. Also there are natural products having this aryl ring chlorinated one. So that means if we can do the arene ring chlorination will be fantastic one, and particularly in the late stage fashion. We thought, okay, let us take our simply this catalyst which I already discussed. It will work, but it did not work. So on this peptide chlorination, so we only got 9% yield. When that we ask why it is not working, the reason is when oxygen, we know oxygen is much more electronegative, so the nucleophilicity is much less. So if we can increase the nucleophilicity, how to do that? By changing this oxygen with the nitrogen one, and when we change oxygen with nitrogen one, these become much more nucleophilic. Even the more challenging NCl bond can be activated. You can see it's a very fast reaction. Within half an hour we got 86% yield, while background reaction is 0. So using this strategy we could do the late stage peptide halogenations, then drugs molecule halogenation or chlorination, actually also bromination we also did using this catalyst. I'll just show you this graph. So when we took without any catalyst, you can see the no reaction even after 12 hours, but when you use that hydrogen catalyst it worked well. And this is the here no induction time. This is much better, much reactive catalyst. So using this catalyst we could do several late stage functionalization of peptide like dipeptide having histidine. Also you can see the background reaction is trace. Yield is little less because of the isolation issue with the histidine. We can use several other, I am not taking the name, like serine and all, okay. So tetrapeptide, even several bioactive peptides, also hexapeptide, even we did also octapeptides, is not I did not list it here. So we can do large number of peptide halogenation. In the background it is the trace, so without catalyst reaction doesn't work. We also use, and for the several drug molecule chlorination it also went well. We not only the chlorination, we can also do the bromination, iodination, and so on. Also several aromatic ring can be functionalized even with the CF3 or nitro group. And if you can see here, we took this dipeptide, we did first chlorination or bromination, vice versa. See, in one pot, just using the same catalyst, adding NBS followed by NCS, it can give you this one with excellent selectivity. So when you do the other one, it either gives some mixture of mono, di, all the things, but here it is very selective. That was another key highlight of the design catalyst. What we found when we did this reaction using hydroxylamine catalyst, it worked in heptane fine. But when you want to do the same reaction in heptane using hydrazide catalyst, it did not work. Then we are checking can we do the computation by DFT calculation. So this is one of my students. We are also working on just checking the mechanism by DFT, at least supporting that. So what we found, when we have like neutral NCl, okay, without hydrogen, we did not get transition state over 6 months after a lot of attempt. Finally, when we put this cationic species, okay, either chlorine can go there or here, you can see they are very close in energy, so they are actually working. So for that, this ionic intermediate or active species, which is actually delivering the chlorine, because they are ionic and the solvent should be more polar like chloroform. For this reaction work in chloroform, but for the previous case, which work like a neutral active species, that work in heptane, that work more like a phase transfer catalyst. So there we could explain why we need chloroform here and heptane there. So you can see without catalyst activation is very high, and here is 16 kilocalorie per mole. So this is for the solvent polarity. We did varieties of reaction, and you can see 114 examples just to show that proof of concept is there. Alright, so while we are working, and we found, of course we know the sulfide is known for the halogenation reactions, but putting several active site on a small catalyst, okay, is a very interesting one. We can make a really active catalyst. So if we check, of course, the artificial enzyme, this is known, but this is a very complex one for us. We also know there are catalysts, organocatalysts, which have the bifunctional one. One is the main active site, some other handle to bind with the substrate, whatever. But the putting several active site on a catalyst and which working perfectly is a very challenging. We do not find any literature report on that. So except the solid state like heterogeneous catalysis. So we thought, okay, it's very easy for us because we are working with the peptide. We took this like methionine, okay, and just couple them, easily synthesize, and then we check its catalytic activity, particularly for the again chlorination. So bromination also to work. So the reaction work with excellent catalyst loading. I'll just discuss in few minutes here. I rarely don't present the table, but here it is for the peptide chemistry, acetonitrile water work. And when you use any secondary alcohol or primary alcohol, they don't work. We know that when you have a sulfide, okay, and at the same time you have the NCH, Kharagpur type of reaction, those are going on. So those organic solvent are not good. It work with several solvent, and chloroform was good, but it work with others also. But terbutanol, because there is no alpha hydrogen to oxidize, so this also a good solvent. You can see with tetrapeptide, catalyst loading can be lowered down to 0.25 mole%. This is quite excellent. So we use this catalyst for the halogenation of several peptides, including the octapeptides, and you can see here, because of we cannot have really lot of products, so like we started with 25 milligram. So this is the reason catalyst loading we have increased. Otherwise catalyst loading are quite excellent, okay, 1.5 mole%. We also did several drug molecule we use for the halogenation, and we could also do bioactive peptides and several heteroaromatic work. So Swimol did this work. This is need to be communicated soon. Alright, now I'll switch the gear. This is about the nucleophilic catalyst design. Now we'll discuss our recent development on the development of organic photoredox catalyst. So we know the organo photoredox catalyst is very important compared to the transition metal catalyst. One is expensive, and of course if we can make the organo photoredox catalyst, which is very cheap, you can make it in 2 grams, that will be the really nice one. So we also started working in this area, and I will tell you our story. So if we check the halide, okay, yesterday Thomas was presenting, like if you take this halide, these are very well known to provide the radical because their potential is much less and it can be easily generate the radical. But the challenging will be the chloride one, most challenging, then bromide, then iodide. these you have to synthesize, but these are very commercially available, you can easily make them. So if you can use them for particularly chlorine and bromine, it will be really nice one for development of several reactions. So they are actually, we took this one, and it would be, of course, it should have low excited state oxidation potential and structure property relationship. We can vary, we can have library of catalysts, we can choose them for our transformation. Alright, so this chemistry started long back. So long before we have developed one on the carbazole chemistry or indole chemistry like this. So it undergo pinnacle type of coupling, and then we get this keto compound, which let the just simply it trap here to give us this benzene relation product. And not only that, this is the mechanism. I am not going to this chemistry today. And if you take this product and then methylate and do the Skoll oxidation, all undergo dehydrogenative coupling. So we got such polyaromatic hydrocarbon with a nitrogen there. So when there is a nitrogen lone pair will be, of course, dispersed among the conjugation, and it make it highly reducing, means it's a electron rich, it can give electron easily. So we choose all of them. Okay, we did the detailed photophysical study and checked it. So what I found, this is difficult to make. So this is a very nice one. We did not take it. So this is very easy to make for us. In 3-4 days we can make it in a good amount. So we call is a benzopyrrolene type of structure, and we have gone through it detailed photophysical study even further. So using cyclic voltagram, so we got the ground state oxidation potential around 0.85 volt against single calomel electrode. Also we measure it absorption and emission spectra, and from the intersection we got the 0 excitation energy around 2.74. From there, using Rayne-Weiler equation, we know it's very we work like this. So this is quite established. So we use and we found that redox potential, this one is around minus 1.89 volt. If we compare the reported catalyst here, ruthenium BP, it is around 0.87, very well known. Iridium minus 1.73, and even CZIPN minus 1.18. So this is quite reducing and easy to make. So we thought, can we use this? And of course it has a lifetime around 4.6 nanosecond, which is okay for us. It doesn't make much difference for our reactivity. So we use these catalysts for several reductive transformation like pinnacle type coupling, immunopinnacle type coupling. I just want to highlight on this point here, if you have the catalyst loading 0.5 mole %, this is really excellent. So this reaction work well. I'll just show few scope. Like you can do the pinnacle coupling 98% it using only 0.5 mole %. The diisopropyl ethylamine we have to use 1 to 2 equivalent, not much, and blue LED is work quite well. This is the catalyst. We can do several. Only thing I found that DR are not good. So when we put really sterically hindered one, the yield little less, but that we got as a single diastereomer. Also we can use the ketone for the pinnacle coupling. We can do the immunopinnacle coupling also, either benzyl protection or doesn't matter. We have several examples I am not showing, so just few of them I just listed out. Another interesting problem, if you think about the peptide chemistry when you are working, you can do solid phase peptide synthesis, but the problem is you have to buy the resin. That's expensive. But if you want to work in a solution phase, we could easily make up to octapeptide. Of course we learn slowly. The problem is when you do the solution phase peptide synthesis, isolation. What we do, we always use the FMOC as a protecting group. Why? Because that is a fluorophore, you can easily detect it. But but when you are not using FMOC, there are not many protecting group which are fluorescent or like UV active. So we thought, can we address that problem, and can we develop a protecting group for peptide which can be deprotected under mild conditions. So we thought, okay, let us work with the sulfone, particularly for the solution phase peptide synthesis. There are many other also fluorescence tag, lot of possibilities are there. So first we choose, peptide case tryptophan is there. So can we put the sulfone on the tryptophan, and histidine or tyrosine. So you can see these our catalyst, BPC catalyst, it again work well with 0.5 mole % catalyst loading only. Okay, and you can see the condition, diisopropyl ethylamine and dry acetonitrile, it work at room temperature. Actually when you shine light, it slowly get warm up. So 35 we choose like the temperature, and you can see naphthyl, phenanthryl, or this, sorry, it will be here actually. So pyrene, all are there, so we can easily deprotect them having different functional group. Even the ketone is there. Ketone are sometimes difficult to handle under this reductive condition, but it can be we can use it. So it doesn't deprotect the aliphatic one, you need the aromatic one. So even you can put two sulfone group, one you can be easily deprotected, which cannot be done under classical method like KOH or methanol. Alright, so the main idea is not to work with those heterocycles, rather working on the peptide. You can see this tetrapeptide, it nicely deprotected. Also hexapeptide, we can have the pyrene, and this again this first time presenting, so this few error I just found. So also here you can see quite complex one. Even you can have the octapeptide, okay, two sulfone group, it can be cleanly removed. Okay, this is very nice method for the peptide sulfone deprotection. Not only that, we can also use it for the like one peptide. You can have a tyrosine protection, you can have the tryptophan protection, and this one tryptophan, tyrosine, histidine, all three are there. Here for every tosyl you have to have 0.5 mole% catalyst loading, so it is 1.5 mole%. All can be deprotected. So we can have the diketopiperazine, bioactive peptide, and also we can have the phenycyl deprotection under these conditions. So this just simply deprotect here, and you can have the tryptophan, and here also phenycyl, all are deprotected. So these are all frozen active, so like their UV active can easily be detected under the by TLC. So these are the how the mechanism work. Actually you see this Stern hallmark plot. So when you take the substrate, it has a more effect, okay, on the quenching, okay, luminescence quenching study. So here it is not much effect, getting not much effect from the diisopropyl ethyl amine. So catalyst get excited, transfer one electron to the substrate, it become radical anion, then it decompose to the sulfone and this radical can undergo heteroatom transfer or takes another electron minus and then take the proton. So this is one way. And also we have done gram scale reactions, and even that time we can, out of 11 mg of catalyst approx, we can isolate 5 mg of the catalyst. So catalyst is very robust, so that's what I want to emphasize. Of course when you use TM4, reaction did not work, probably the radical is the intermediate. So after developing this catalyst, we thought, okay, this is needing 4 step. Can we go even better catalyst? Okay, so then the speaker says we did not expect this will be also a beautiful catalyst. I am not going details about their photophysical properties. So we did the same way, like UV visible, fluorescence, then we got the cyclic voltammetry. All together we have done a detailed study of several series of catalysts, and we have shown only 5 of them. So they are really reducing, and the highest reduction potential, it becomes even more, minus 2.07. Now we are working on them and we have even better. I think we are not sure, we have to do in globe box because of the anionic one, and we think it is going 2.8 minus. So it will be really good for the chlorine activation or the aromatic substrate which are electron rich. So we are keep on working in this direction, and you can see in the HOMO by DFT we calculated, you can see here the HOMO or LUMO almost dispersed throughout this this benzo-carbazole ring, and for the HOMO C7 ring has some contribution, C8 is 0. For the LUMO one, this is 0, this is little contribution. So this is not a donor acceptor type, it's like a throughout the molecule it is distributed. That is what we want to say. Alright, using this catalyst, and we can easily do several transformation like peptide desulfonylation, as I mentioned. We have done few example there. We could also do this radical cyclization, like yesterday we have a nice talk on that on sp3, but it is sp2, it coupled there through radical intermediate, and we can have a very good scope. And when it doesn't migrate, I'll discuss the mechanism in the next slide. So you can quench it here, we can also get such product. So reaction also can be done in gram scale, having the good functional group tolerance. So how it work? Again, the actually catalyst giving electron to the substrate to get us this radical. You can see from this plot. I'm not going here, and it generate this intermediate, then aryl get transferred there to give this one, or here it can undergo single electrons transfer followed by protonation to get this dearomatized product, or it can go transfer like here, it migrate, then aromatization will give you this cyclized product. We got some other nice product also which just we are working, so we are completing on the scope. So it's not only on the desulfonylation or CC coupling reaction, we can do the borylation. There is we have a several scope, like around 18 scope. You can do the diborylation. You can use the chlorine, bromine, iodine. Chlorine case is yield is less. We need to work more on this, particularly maybe the new other catalyst design. So we really want to activate the chlorine. Bromine is always best for us. The chlorine will be the next target with the other catalyst. We can also do the phosphorylation. We can add these phosphates. So even with NH free is very challenging. Several times it doesn't work with the NH free, but still it work here with the indole, with the benzyl one, so it work well. We can also do the pinnacle type of coupling, immunopinnacle type of coupling, similar set of reaction, very much possible. Not only that, I came to the last part of my talk, so I have at least 2 more minutes. So we also work on another catalyst we call the this orthoquinone. We know in the enzyme cofactor in the mother nature, so how they oxidize the amine is just using this orthoquinone. Okay, so this is connected to the artificial, like in the lab, artificial proteins or so in the mother nature also. So this chemistry is nothing like it is there since 1990s by Vignan. Then it slowly saw the gradual improvement and until here. And while we are working in this area, we found these actually made several such carboxyquinoline alkaloids. These are alkaloids, natural product. We made a natural product. We asked this question: okay, whether these such molecule in nature also working like this. So that's the reason actually idea came. So we wanted to this methyl, if you put, this is natural product. We have made good number of them, and then we saw that okay, this should also work. And after some study, we could do this such coupling reaction, not coupling, actually oxidation followed by imine formation, C base formation. The reaction are quite high yielding. Catalyst loading was 10 mole%. We can also do the cross coupling, so such reaction are possible using this catalyst. I will not go much about the DFT calculation. Only one thing I want to say. So this is nice observation. When your student work and he does the DFT, he knows what going on. We discuss so extensively, so many mechanisms are possible. Finally, we found intramolecular hydrogen transfer. This amine assisted in the transition state. Not only that, when this phenyl, whether in the pseudoaxial or pseudoequatorial, that much also give you quite a lot of difference in terms of activation energy. So this is TS2 equatorial. This is the real transition state as supported by GFT. All right. So yeah. So what I will summarize. So if you what we started with this catalyst, first generation catalyst, then we moved to the more reactive one, then this one, okay, with the multiple active side. Then we have showed you that BPC is a beautiful catalyst, but if you want to really use it, this is damn cheap. So it will take 2 to 3 days, and you can get in grams, 2 grams also. Okay, it's very easy to make it. No metal in, it's just no expensive reagent, I would say. So this is also working well for amine oxidation. These are the organocatalysts we recently developed from our laboratory. Alright, so this is IIT Kharagpur main entrance. This is the main building, and our lab is here on the 6th floor. So our office is just opposite to this. I think Thomas and Tatiana, they visited in just downstairs opposite sides. We have a big building, and our infrastructure also beautiful. So please do visit us. I'm inviting you. And I just one picture with the group. So inside the lab, student while working, sometimes parties, we go for excursion, all are there. In addition, yeah, so I like to thank all the funding agency, and thank you all for being here.
Thank you very much for the wonderful talk. So open for the discussion.
Very nice talk, thank you.
Thank you.
I have 2 questions. The first one, when you use your catalyst to reduce the tosyl imine, right? So are you sure that this is going to be a set mechanism, not energy transfer, and you have 2 radicals?
It is known actually the set single electron transfer, then it undergo like your pinnacle coupling. You're talking about the imine dimerization. Yes. This type of reactions, like the pinnacle coupling, that is the mechanism what
but you have
accepted already.
You have NS bond, and there is a lot reported in the literature that it undergo through energy transfer mechanism.
So for the imine reduction, whatever, actually it is not that we are proposing. This immunopinnacle reaction is quite well known. It is reported to go by electron transfer, then radical anion, they couple. So that is reported.
So you're using amine. So do you think that it's a XRT process, or so you are abstracting? How do you abstract the chlorine exactly?
That's a good question. So I think electron goes to the aromatic ring, then it undergo decomposition, Cl minus goes away. I'll take, like, even the sulfone reproduction also work. Like sulfone take the electron, the aromatic ring. That is the reason it work with the aromatic, not the aliphatic sulfone.
So you don't think that the oxidized amine play a role to directly abstract the chlorine or the halide?
No, I don't think so. I think it goes to the ring and then it decomposes. And for sulfone, there is a proof that whenever methyl group in place of any aromatic group, it doesn't work at all. It's untouched.
Thank you.
Yeah, electron always goes to the pi star orbital. Yeah.
Thank you very much for this very nice talk. So I have two questions about the your first part. So between your first generation and second generation of catalyst, you have in one case you have an alkyl chain, and in another case you have this aromatic ring with a therapeutic substituent. You didn't comment about the influence of this.
Yeah, actually this is the proof of concept. First of all, the alkyl chain, when we put, just to make it more soluble. So if you see, when you are putting the phenyl group, yield is 70%. It's not the catalyst problem, rather the solubility problem. So we put the para-tertiary butyl group, then it becomes again soluble. So one may ask why to put the aromatic and why to aliphatic. Aliphatic for achiral catalyst, perfect. Okay, but our idea is not the achiral catalyst, going for asymmetric. So we have to, aromatic has to be there. Yeah, we're working on.
It was my second question. So if you want to do an asymmetric version, do you think that you can have, instead of a sulfonamide, a sulfinamide chiral 1 SU instead of SO2?
Actually, sulfinamide possible, but sulfinamide are known to undergo oxidation. Okay, like Jeff was presenting, and we are also working a lot on the sulfinamide chemistry. So that are known to under oxidation, but we don't need sulfinamide. Sulfone only. We have made some catalysts already, chiral catalyst. So those are going on. So yeah, I hope some result will come in the coming days.
Okay, thank you.
Yeah.
Thank you, Modhu, for this great talk. Sorry, I've been a bit late at the beginning.
Yeah.
So I have actually two questions. One which is related to Annie's question. So your NC catalyst, when you showed us the absorption spectrum, you have two wavelengths of absorption, two maximum of absorption. So do you have any idea why? Because it's really
that question we got also some expert from physical chemistry who really work on these absorptions. So I don't know, like it can be the aggregation, means the real structure we don't know in the solution what actually. So there is 370 and 390.
Yeah.
So 391 we choose as a purple light and went ahead with the work, like with the catalysis, it worked fine. But as you mentioned, why 2? Honestly, we don't have answer at the moment. How it is coming?
Okay, because I've been really intrigued by the reduction of aryl chloride, because when you showed us the physical-chemical properties of the catalyst, it does not really match with the redox potential of the aryl chloride. So do you think that you might get actually another active species where you pump an electron into the structure and you made a radical anion which is photoactive, and you photoactivate the radical anion which is then able to reduce the aryl chloride?
Yes, actually what you are saying, that is what we did the stand forward to. So here we have done not only one substrate, several substrate. It always, so catalyst, if you want to take electron, there is only diisopropyl ethylamine. It doesn't have much effect. It always the substrate which quench the catalyst. Okay, so that means the catalyst giving electron. Now, as you mentioned, the potential, I agree with you, and chlorine are not that good at yielding. Okay, so the yield is maybe 37% we have. Bromine is working fine, which is matched in that zone. But with the chlorine one, yes, possible that in the system the catalyst is not a discrete molecule. So altogether, what actually they are doing probably is a big question.
So perhaps you can pump an electron from another catalyst into the catalyst, or from the substrate to the catalyst, and you made an active catalyst.
I would say from the substrate to catalyst will be difficult. However, for the catalyst to catalyst, I am not sure. Means I don't have any data or any observation to support at the moment.
Because perhaps you can try to photo irradiate your CV and to see what's going on.
Yeah, that actually those things we did not do it. So maybe
Because you have some kind of pyrene, and pyrene are well known, to be a photoactive radical anion.
yeah, only for pyrene there means one can ask question. The pyrene itself can be catalyst.
Yeah.
Okay, I agree with you. So that's the reason we always did the background reaction for the pyrene. If you expose, to be honest, if you expose that substrate without catalyst for fairly long time, maybe let's say 2 days, you may get 50% yield, but you won't get within 1 hour or 2 hours. So pyrene itself does catalysis, not other substrate, only for the pyrene case. But as I told, here catalysis is involved, but pyrene also can give you the background reaction. Okay, but take a lot of time, efficiency is much less. Pyrrolene, yes, pyrrolene is known, 10% probably catalyst loading. Pyrrolene also known for as a catalyst, but the catalyst loading is very poor, like 10%. Here is 0.5.
I really like your 2-generation catalyst, and then I was wondering what is the efficiency compared to the amide version. Do you have any idea?
Yes, we also not only NH2, we did with the NH tarbutyle, so it gives around 50% yield. So amide is also a nucleophilic catalyst, but the efficiency much less. So reactivity of this hydrazide are super. So that result I must have added. So with NH2 0, when you put alkyl group around 50, and this one is almost quantitative. Yeah, we consider that. Honestly, the acid part, this work we have to do. Not only sulfone, there are many acid, because when you are moving toward the asymmetric synthesis, that's very important. Yeah, thank you. I think we have to do some work. Yeah, thank you.
Okay, just a small because the pHs are solubility very difficult. How about solubility and stability of these compounds?
These are very stable, and solubility is quite nice, because you can see this is not a planar one. Okay, this is like a helicine type. Here is the helical axis. So these are all quite soluble. So solubility not at all a problem. Okay, of course, if you want to do this reaction in toluene, the problem the catalyst solubility, rather than NCS doesn't soluble. So that's the problem. Stability is super. Stability not a problem. Even after 5 mole percent, 0.5 mole percent catalyst loading, still we can recover 40% of the catalyst. So that is quite nice. That's the advantage.
So thank you very much for the talk.
Thank you.
Please join me once again, and thank you for the excellent presentation.
Thank you. Thank you very much.
Long live India and France friendship.